Hey guys, just had a hey,
Welcome to the second week of Introduction to Bioengineering. This week, we're going to talk about innovations in medicine. We're going to talk about how bioengineers can help develop new medicines and can scale the production of medicines in order to ensure that there's enough for everyone. There are 4 learning objectives for this week. The first is just to understand the process by which new medicines are developed. We want you to think like a bioengineer and to see if you can think of ways of developing new medicines by telling you about the process. Then we want you to describe the difference between categories of medicines. This week, we're going to talk about small molecule-based medicines, protein-based medicines, and whole cell medicines. We want you to understand the difference between these medicines and the ways that bioengineers can help create new medicines of all 3 types. We also want you to understand the importance of protein structure in how well a protein-based medicine functions, and then to understand the tools available for measuring and predicting protein structure. And before we get into this week, I want to pose a simple question. What is a medicine? Maybe when I ask this question, you think of things like this. These are examples of drugs that you can get at the pharmacy or via prescription from your doctor. The Food and Drug Administration has a specific definition of a drug, which I'd like to go over so that we can all be on the same page as we enter this week. The Food and Drug Administration defines a medicine as a substance that's intended to be used for the diagnosis, cure, mitigation, treatment, or prevention of disease. So it's something that you need to take in order to feel better or diagnose a disease. It's not a food and it's not a device, but all other things can fall into the category of a medicine. Not a food, not a device. Okay, so we're going to discuss 3 categories of medicine this week. Like I said, those are small molecule drugs. These are molecules that can enter cells very easily because they're very small or they have a low molecular weight. These molecules typically affect the function of proteins within a cell, and that is how they cause changes that treat disease. There are also protein-based medicines. Proteins are either these protein-based drugs are either of naturally occurring proteins or ones that have been modified, and the proteins are administered to people in order to help treat disease. And then lastly, the whole cell medicines are living cells that are administered to people to treat disease. Sometimes these cells are engineered in order to perform a specific function within the body, and sometimes they're just naturally occurring organisms that we don't have enough of within us at any given time. Think like probiotics, but to treat disease. Okay, so the 3 steps to developing new medicines that I want us to focus on are identifying potential medicines. So that's the first step in the development of something new, testing to see whether or not these medicines work, and then scaling up production. And our goal for the week is to think about how you might improve or accelerate each step. So I want you to be thinking throughout the week, I want you to be thinking like, where can I look for new medicines? How do I know if this new thing that I've identified is working? Is it actually helping to cure someone's disease? And how can I make enough of a medicine once I know it works? To sort of frame this discussion, we're going to have 3 different case studies. We're going to talk about syphilis, malaria, and diabetes, and the drugs that are available to treat each of these diseases. We're going to go back in history and see when the first treatments for these diseases were developed. And as we go through this, I want you to pay attention to what observations people made in order to come up with a new treatment idea. So somebody had maybe a friend who had syphilis, and they're like, what do I do to treat this person? Well, they observed something around them that led to a hypothesis about what they could use to treat their friend. And so I want you to keep track of what the observations are that led to new treatments. I also want you to pay attention to the methods that were used to figure out if the new medicine worked, right? It's one thing to have an idea for how to treat a disease and another for it to actually be functional. So how do we know if the medicines work? And then how was production of the medicine scaled up to treat everyone in need? Those are the three steps in developing medicines, explained slightly differently. And so I want you to pay attention to all three of these things as we go through the week. I'm excited to talk to you about syphilis, malaria, diabetes, and protein-based drugs. So hopefully this week is an exciting module for both you and me. All right, bye. Today we're going to talk about the discovery. Today, we're going to talk about the discovery of drugs to treat syphilis and malaria. And as we go through this lecture, what I want you to do is practice thinking like a bioengineer that's trying to develop a new medicine. I want you to observe how other people have done it in the past. As we go through these stories, think about how people identify new potential medicines, how they test to see if those medicines work, and then how they scale up production. Because these are each of the areas where bioengineering is really having an impact on new medicine development. So after we talk about how it's been done in the past, we'll talk about all of the new things that bioengineers are doing to accelerate each of these steps in medicine development. Before we get to the medicines, we're going to talk about the diseases. I want to introduce you to malaria and syphilis. Hopefully they're not super familiar to you yet, but malaria is a very common disease, actually, so maybe it is. Malaria is transmitted from person to person by mosquitoes, and when it is infecting you, it causes fever and chills, abdominal pain, chest pain, headache, nausea, and vomiting. And it's still really prevalent. So this is just a map of malaria infections globally in 2019. And you can see that there are areas where there's more than 400, almost half of the people have been infected with malaria in 1 year. Now, in 2020, globally, that looked like 240 million cases of malaria and 627,000 deaths. So this is a very serious disease. And it's caused by a parasite. So malaria is caused by a parasite called Plasmodium falciparum. And that parasite lives for part of its life inside human red blood cells. So on this screen, you're seeing red blood cells. Those are these kind of big, light purple balls. And inside of them, or at least some of them, are malaria parasites, these darker purple stains. Now, living inside of a red blood cell is just one stage of the malaria life cycle, but it is the stage that causes symptoms. So here's the full life cycle. Like I mentioned before, malaria is transmitted from person to person by mosquitoes. So when a mosquito bites you, it delivers parasite into the person's bloodstream, not on purpose, but just because the process of biting pushes some material from the mosquito into the person before they start sucking up blood. Okay, so once those parasites are in your bloodstream, they'll find their way to your liver, and there they'll get into liver cells and make copies of themselves. And then they'll bud off into vesicles, and those vesicles will enable the infection of red blood cells. And like I said before, it's at this point when the malaria parasite is infecting your red blood cells that you start to have symptoms. And then eventually the red blood cells will burst and you'll get parasite in your blood that can be sucked up by a new mosquito and delivered to another person. There's an important aspect of malaria's or Plasmodium falciparum's life cycle that occurs inside of the mosquito. I'm not going to talk about it today, but I encourage you to look it up. It's really fascinating. Okay, so this is malaria. It's caused by that parasite, Plasmodium falciparum. Syphilis is a sexually transmitted disease. It's caused by a bacteria, and its symptoms occur in 3 stages. In the first stage, which occurs 3 to 90 days after exposure, you get a sore. That sore appears either in your mouth or on your genitals, but it's not painful. Eventually, the sore heals, and about 4 to 10 weeks later, you get a rash. And that's a full body rash. And for some people, the syphilis infection stops there. They never have another symptom, and they live the rest of their life normally. But for other people, about 3 to 15 years after the initial infection, their syphilis disease reappears, and this time it's much more severe. Stage 3 syphilis affects your internal organs. It's actually lethal. It causes dementia and other psychotic disorders. It will cause internal bleeding, like I said, damage to your internal organs, and respiratory failure. Here's actually a photo of a skull from somebody who had syphilis. And you can see the degeneration, the holes within it that are caused by the disease. Now, syphilis is caused by a bacteria called treponema. It's not infecting red blood cells. It actually doesn't live inside of your cells at all, but it is what causes syphilis. So I want to ask you, after hearing about these 2 diseases, how would you cure them? What would you do to treat somebody who has malaria and syphilis? Well, what would you do if it were the 1800s? Imagine there are no antibiotics. We don't know about bacteria. We don't know about plasmodium. We really don't know what's causing these diseases, but we know that people are getting sick. We know that they're dying. What would you do to try to cure these diseases? Take a minute to brainstorm. Maybe you're lost. Maybe you have an idea. Either of those are okay. But check in. See how you're thinking about this problem. All right. Now, what actually happened? Well, a cure for syphilis started with an observation. We're going to talk about the cure for syphilis first, and then we'll get to cure for malaria. This observation was made at a mental hospital. Like I said, syphilis can cause neurodegeneration, and so a lot of patients with stage 3 syphilis would end up in mental hospitals. And a doctor at the mental hospital noticed that syphilitic patients occasionally were cured by what he called febrile episodes. These are fevers. And so he thought to himself, maybe a fever can cure syphilis. Didn't know why that would work, but he thought it was an idea worth testing. So the challenge became, if a fever can cure syphilis, how can you induce a fever? This question of how could you induce a fever is not an easy one to answer. People tried wrapping people up in blankets. They tried doing all sorts of physical things to raise their temperature. What worked really well was actually infecting patients with malaria. So if you'll remember, malaria induces a high fever. It's one of the symptoms of having Plasmodium falciparum in your blood. And so this doctor, Dr. Wagner-Jorig, the one who made that observation in the psychiatric hospital, tested Plasmodium falciparum as a treatment for syphilis. He extracted malaria parasites from a patient who currently had malaria, and then infected a stage 3 syphilitic patient with that malaria parasite, with Plasmodium falciparum. And this is risky, right? Like, what if you're wrong? What if the fever isn't strong enough to kill syphilis? What if it wasn't actually the fever at all that was causing patients in the mental hospital to recover from syphilis on occasion? But Dr. Wagner-Jorig got lucky, or rather, he knew what he was doing, and it worked. He won the Nobel Prize for this work, actually. for discovering the therapeutic value of malaria in the treatment of what, in this case, they're calling dementia paralytica, but is actually syphilis. So just to be really explicit about it, the treatment that Dr. Wagner-Jauregg prescribed or started using to treat syphilitic patients looked like this. There'd be a patient who had stage 3 syphilis. It's important to note here that he was only treating stage 3 syphilitic patients, right? At stage 2, you just have a body rash. The disease is not that lethal, and it's possible that you'll never develop stage 3. But once you start showing stage 3 symptoms, it's pretty bad. So he would infect these stage 3 syphilitic patients with malaria in order to try to cure them, and then wait for the malaria parasite to induce a fever in person. After the fever, hopefully their syphilis, the bacteria that causes that, was dead, but the malaria parasite would still be very much alive within the person. And so the only reason why this was a viable treatment approach was because a cure for malaria already existed. There was something called quinine that was being used in order to treat malaria. And so once the patients spiked a fever that cured them of syphilis, he would administer quinine and cure the patient's malaria too. Okay, so here's just a slide saying that explicitly. This P. falciparum, Plasmodium falciparum treatment only works if you can cure the malaria that it causes. Otherwise, you're just swapping one disease for another. You're trading syphilis for malaria, which isn't really better. Okay, so that begs the question, how did a cure for malaria get found? Malaria had been around for quite a long time. And like the cure for syphilis, a cure for malaria started with an observation. The Quechua people who were living in what is presently Peru were using bark from a cinchona tree, a certain type of tree, in order to treat fevers. And this was noticed by a Jesuit colonist who had come over from Italy or Spain and realized, that there were, or come over from Spain where there was a lot of malaria, and thought, hey, if this cinchona tree bark can be used to treat fevers, maybe it can be used to treat that fever that's going around in Spain. And so he sent a sample of the cinchona tree bark back to Spain. And then for 300 years, cinchona bark is shipped from Peru to Europe to treat malaria. It worked, I should say. And no one really knew why it was that this bark was able to cure people of malaria. They just knew that it worked. And so they have the Quechua people to thank for the cure to malaria. Okay, so notice that it's 1630 when this Jesuit apothecary first sends cinchona tree bark to Europe, and that it took until 1820 to isolate quinine as the active component of bark. People didn't know for a really long time what it was about the bark that was making it a useful malaria treatment, but eventually they figured out that it contained a lot of this molecule called quinine. Once the population in Europe grew and there was more demand for quinine, more cinchona plantations were planted. And this time they were planted not in Peru, but in Indonesia, which also had a hospitable climate for this tree. So, for 300 years, Peru is the source of all of this bark. And then from 1910 to around 1944, this bark is being supplied from a plantation in Indonesia. Okay. In 1944, in 1939, World War II breaks out, and the Allied nations are cut off from their supply of cinchona bark. And this is a problem because of a lot of fighting, and a lot of fighting in World War II was happening in malaria-heavy regions, in regions where there was a lot of parasite and a lot of potential for soldiers to contract malaria. But when they're cut off from the plantations, when they're cut off from the source of this bark, they have no treatment options for malaria. So this war sparks a massive hunt for cinchona bark alternatives. People are trying really hard to synthesize quinine in the lab so that they don't need to get it from the bark of this tree, but they have no idea how to put this molecule together. And so they start doing a lot of synthetic chemistry to figure out if they can get this molecule made in the lab. But they also start screening other alternatives. So they have libraries of chemicals that serve as dyes for like the clothing industry and other textiles. And they think, okay, well, we have all of these compounds, we have all of these molecules. Maybe one of them is also effective against malaria. So scientists start digging out every chemical they can think of. They go through these large libraries to see, do we have something on hand that can treat malaria? And what they find, when they're doing this screening, they're actually testing for efficacy against malaria in birds. Turns out birds can get a form of malaria that's very similar to the form that affects humans. And so you can see whether a potential treatment for humans might work by giving malaria-infected birds whatever compound you think might function well as a medicine. Okay, so they're testing all these different dye compounds. They're trying to modify the compounds that they have in order to make new potential medicines. And what they end up with is something called chloroquine. Chloroquine is a synthetic alternative to quinine, and it becomes the dominant anti-malaria medicine, not just during the war, but actually for quite a while after. And that's in part because of resistance to quinine and in part because of the ease of synthesizing chloroquine relative to the extraction of quinine from cinchona tree bark. Okay, but eventually the malaria parasites, Plasmodium falciparum, start becoming resistant to quinine. And this is sort of similar to what's happening now with antibiotics. We see that there are bacteria that are resistant to our first line antibiotics, like penicillin, things that we would usually administer at the first line of an infection don't work on bacteria. They're starting to evolve. And so the malaria parasites at this time, starting around the 1950s, but really accelerating through the later half of the 1900s, we start to see a rise in chloroquine-resistant malaria. And so this is a problem. If chloroquine is your method of choice for curing malaria, and chloroquine doesn't work on an increasingly large fraction of the parasites that cause malaria, then you need to find a new treatment. And one of the places where this was very important was in China, where there was a large population of chloroquine-resistant Plasmodium falciparum, where there was a lot of malaria that could not be killed with chloroquine. And so the Chinese government instructs scientists to start to hunt for an alternative to malaria. And, by 1969, there are more than 240,000 compounds that have been screened for anti-malarial activity worldwide. And they haven't found stuff that's better than chloroquine and quinine. But this Chinese scientist, Tu Yu, begins screening Chinese herbs, remedies that have been used for a long time in China to treat different ailments, but have not been tested by Western scientists against malaria as a potential treatment. And so she starts screening all of these different herbs. And at this point, we can actually screen for red blood cells in a dish. So we don't have to, she's not doing experiments with malaria infected birds. Instead, she has samples of red blood cells that are infected with malaria, which she can then treat with these different herbs by kind of grinding them up and adding them to the cells and seeing whether or not this kind of treatment cures or kills the parasite, but leaves the red blood cell relatively intact. So through these types of screening, she identifies artemisinin. It's a compound from a shrub called Artemisia annua that kills the malaria parasite faster and in all life cycle stages. So that's really exciting. So once she discovers this through her cell-based screens, they start clinical trials in people and discover that not only does it work well at curing malaria, it has fewer side effects than chloroquine or quinine. And so Tu Youyou gets the Nobel Prize for Medicine in 2015 for this discovery. And that's a very exciting time because we have now a new and even more effective treatment for malaria, which is still a problematic disease worldwide. But it's actually difficult to source large quantities of artemisinin. In this big shrubby plant, artemisinin content is only 0.1 to 1%. So you have to grind up a lot of the plant in order to get enough artemisinin to treat people. And it's not just that artemisinin crops are, exposed to the weather. And if there's a bad crop year, then you can have less medicine and it can change the prices of this anti-malarial drug in a way that pushes it out of reach for people who might really need it. So this is an area where bioengineers step in. They see an opportunity to produce this compound, artemisinin, in a different way. They think, why don't we try engineering yeast to produce this compound instead? Then we can move from the field where conditions are variable to fermenters where we control everything. Okay, now how do you do that? How do you engineer a bacteria to produce this medicinal compound that's usually found in a shrub? Well, you need some background information. You have to know how the plant cells, the cells of that Artemisia annua shrub, produce artemisinin. And fortunately, there had been a lot of work on plant genetics to figure out exactly which genes encoded in Artemisia annua's genome lead to the production of artemisinin. So this is just showing you all of the compounds that are intermediates as you move from acetyl-CoA, which is a part of central metabolism, all the way to artemisinin. And in bold uppercase letters are the genes encoding enzymes that are responsible for making artemisinin from acetyl-CoA. And what you'll notice on this slide is that there are a lot of steps to go from acetyl-CoA to artemisinin. And there are a lot of genes that do those chemical reactions. And so it's not going to be an easy feat to try to move all of these genes from this plant into yeast. But there's a researcher at Berkeley who really wants to try. So Professor Jay Kiesling at UC Berkeley initiates a project in his lab to express all of the enzymes necessary for artemisinin production in yeast. And he's able to achieve 25 grams per liter production of this compound in yeast just by putting these genes from the plant into yeast. And this is really exciting to people who work in bioengineering, and it's an example of new technology being used to try to change the way that medicine scale-up occurs. But there's some market resistance. So there are people who are maybe not as happy with the use of genetically engineered yeast to make medicines because it's not making a whole lot at first. That titer, although pretty good, could be higher, and the process of producing artemisinin in yeast could yield more artemisinin per cell. And
And people were excited to kind of see it there, and were maybe a little disappointed at first at the yields, but also, there are social factors at play here, where people whose whole livelihood was to farm this shrub in order to produce artemisinin were going to potentially be put out of a job by bioengineers who had taken their whole process and moved it into a fermenter. And so there were some kinks to work out, but the kind of financial drivers, this need to produce low-cost medicines, really got the Gates Foundation involved, which helped push scale up to see if we can produce 25 grams per liter of artemisinin in the lab, how much can we produce when we start moving these engineered yeast into bigger and bigger bioreactors? I am listing a bunch of news articles here, mostly because I would encourage you to read them, to see kind of how this new bioengineering invention was sort of received, and how it's kind of found its way to where it is now, where artemisinin is being, yeast-produced artemisinin is really being much more heavily embraced and pointed to as a great innovation in bioengineering in solving a key problem in medicine, which is scale up, or contributing a solution, I should say. Okay, so those are treatments those are the stories of the development of medicines to treat syphilis and malaria. And I just want to put them into context for you. So remember, at the beginning of this lecture, I said that I wanted you to pay attention to the steps to creating new medicines. Remember, we need to identify a potential medicine, test to see if it works, and then scale up production. Well, if we think of Plasmodium falciparum, the parasite that causes malaria, as a new medicine, then this first step, identify a potential medicine, occurs when someone observes fever-reducing that a fever reduces syphilitic symptoms, right? And then that leads them to test Plasmodium falciparum as a fever inducer, right? So they think, okay, a fever could be used to treat syphilis, and I know that Plasmodium falciparum causes a pretty high fever, and so I'm going to test it as a fever inducer. The scale up in this case is sort of weird. The doctors were sourcing parasites from malaria patients. This is certainly not as widely adopted of a practice as any of the other drugs that we talked about today. But it is how this particular treatment was sourced. So I wanted to put it in here. Maybe you can think of a better way to source Plasmodium falciparum for the treatment of syphilis. Okay. So actually, I do want to just say that we don't treat syphilis with Plasmodium falciparum today. Syphilis is caused by a bacterium, and we have antibiotics that can kill it. And so if you were to contract syphilis today, you would get an antibiotic from your doctor, and that would cure you. Please don't seek out malaria as a treatment for a disease that's much easier to cure today than it was in the 1850s. Okay, so now let's think about cinchona bark, or quinine as a new medicine. So when colonialists observed the Quechua people treating fevers with cinchona bark, that led them to the hypothesis that cinchona bark can be used to treat malaria, because malaria also has fever as one of its symptoms. They sent cinchona bark back to Europe to be tested as a treatment against malaria. I should say the fact that cinchona bark was being the diseases that the Quechuan people were treating with cinchona bark were not malaria. It just was fortunate that this compound happened, or this bark happened to work against malaria and the other ailments that the Quechuan people were treating. I don't want you to think that they were already treating malaria. Okay, so they sent this bark back to Europe as a treatment. So that's how they were testing to see if it worked with people. And then to scale up production, they decided to create large plantations of cinchona trees. Now, that is problematic, and there are better ways to produce a lot of medicine. I think bioengineering can have an impact in streamlining these processes of medicinal development and scale up, and can avoid situations where we feel like we need to plant very large quantities of a tree, for example, in order to get a lot of medicine. Okay. Chloroquine as a new medicine. Here, the identifying a potential medicine and testing to see if it worked was these two steps are being done simultaneously. So here, scientists are screening a large chemical library of dyes for the ability to cure birds of malaria. And so it didn't necessarily spawn from a fortunate observation, but from a directed screen. And then they scaled up production through chemical synthesis because they already knew they could synthesize this molecule. And then finally, we have artemisinin as a new medicine. This was also a situation where the screen and the testing to see whether or not it works were kind of hand in hand. But I want you to think about how you might innovate in each of these areas, and how the advancement of science, our ability to have models of diseases and to engineer organisms to produce a lot of compounds, really changed the way we search for new medicines and the way we scale up production. But just because we have new methods of doing this doesn't mean that some of those old school observational methods are worthless. It's still worth looking around you to see if there are things about the world that you observe that might be or lead to a new medicine sometime in the future.
All right, welcome back.
Today, that you can observe that might be or lead to a new medicine sometime in the future.